Hello friends, welcome to Science Jagat. Today we are going to talk about detection and isolation of somaclonal variants. So before going towards detection and isolation, we should know what is somaclonal variation. I have already made a detailed video about it, so you can go to this channel Science Jagat and search for it. And for now, I just give you a crisp glance of somaclonal variation. So Soma means somatic cells, clonal means clones, that is generations, and variation means changes. So changes in somatic cells which carry on from generation after generation, that is somaclonal variations. Genetic variations in plants that have been introduced by plant tissue culture and can be detected as a genetic or phenotypic trait is called as somaclonal variations. Okay, now isolation of somaclonal variants. This can be done by two processes, that is without in vitro selection or with in vitro selection. So first let's see what is without in vitro selection. In this kind of selection, you will take an explant. An explant can be a leaf, stem or root, etc. And then we'll culture it in a suitable media. Suitable media means you will take the media and the growth hormones according to the explant you are taking, right? Okay. Then we'll supplement that suitable media with growth regulators. After a few subculturing, you'll see a callus growth in that media. Okay, so that callus is an unorganized mass of cell which is developed from the differentiation of explant cells. Okay, now we will transfer that cell into uh, another selective media that contains shoot induction medium. Okay, that contains some growth hormones which are required for shoot regeneration. So till now, this media does not have any kind of selective agent or any toxic inhibitor or inhibitory substance. Fine. Now, when we give time and after subculturing, that shoot will regenerate in the media from that explant. Okay. Now, the plant produced are grown in the pot and then that would be transferred to the field so that it would get acclimatized and no, it will get habitual for the soil environment. Then we'll go for the screening of the desired traits, then the selection of somaclonal variants, that if that plant that the shoot regenerated, that plant regrown have that special characteristics which we are searching for, right? And then that plant which is selected as a somaclonal variant will go for agronomic trials. Okay, so this is without in vitro selection. We are not using any kind of selective agent here. Okay, now let's see what are the limitations of without in vitro selection. So there is no specific approach for isolation of soma clones, as we are not using any specific toxic agent, or we are not checking out any specific microorganism for resistance in the plant. So we are not so sure that what kind of variation the plant have. The next one is the appearance of a desired trait is purely by chance. So if the variation in the soma clone is for a physical trait, then only we can check it by the appearance. Otherwise, if there is a variation which does not appear physically, or it could be a protein, it could be a molecular base variation, so we cannot no, check it by the appearance. Third one is the procedure is time consuming and require a screening of many plants. So as we are not much specific for the selection process, so it is of course very time consuming, and we have to check the somaclonal variation traits in many generations and many number of plants. Okay, so this is time taking. Now let's see what is with in vitro selection. Here, isolation of soma clone with in vitro selection method basically involves handling of plant cells in culture, we can do culture of protoplast, callus culture, or any other explant. Like we use selective agents for this, like microorganisms or selection of biochemical mutants or specific toxic agent to check the resistant cells. Okay, so let's see what is the process. Here we are taking a plant, we take out an explant from that, and we allow that explant to grow in a culture media and to grow it in a callus. Sorry. Now we'll subculture this callus and allow it to proliferate, and maintenance of culture will be done by subculturing it again and again. Now, on the another hand, we are using a pathogenic organism. We'll culture that microorganism, make a purify culture of that microorganism, and then we'll isolate the particular toxin we are searching to check the resistance in the plant. Now we'll use that toxic agent and add it in the media where we are growing that calli. Fine. So now the culture which have the callus also contains a toxic agent for which we are testing if the callus cells are resistant for it. Right. Now after many subculturing cycles, isolation of tolerant cells will be done. How it is so? The cells which are resistant to the toxic agent will survive and others will be dead. Fine. So we'll take out the survived cells and go for subculturing in the specified media. Now we'll regenerate by allowing that those cells, selected cells, to grow in shoot induction media, and then a plantlet would be grown. Then we'll again test it for the pathogenic toxicity. Now what we'll do, we'll grow those cells in the selective media and we'll add that microorganisms in that media and check if the cells are showing resistance for that particular microorganism which is making the selective toxin, right? And we'll do this for generations, for first generation. Then we'll take that plant to the pot and allow it to get acclimatized in the soil, and then the plant will start self-pollinating, or vegetative propagation would happen. Then after progeny after progeny, we'll check that those plants have those resistance power or not. Then we'll test for disease resistance, and then disease resistance would be checked for at least 2 generations more. Then we'll take that plant to the agronomic trials. Okay, so this is what we are doing in vitro selection. We are using a particular selective agent, and then we are subculturing the survived plants which have resistant due to somaclonal variation, and then we are checking them for the particular disease resistance, and then we are taking it for the agronomic trials. So here lies the major difference between both of them. Okay, so there is one more example for somaclonal variation in improvement of agriculture crops, and this is by in vitro selection. So as I told you, we are taking a callus or a tissue or an organ from a plant, exposure to selective agent. Here we are using NaCl because we are learning an example for salt tolerance. Before that, we have learned about toxicity of microorganism, right? Now we are taking salt tolerance as an example. Now, after exposure of selective agent to the callus, we subculture the survived callus in the selective media, which contains NaCl. We'll subculture it till 3 to 4 times, right? Now, non-tolerant cells of the callus will be discarded. The selected NaCl tolerant cells will be selected, and will grow them in this again selective media and perform certain physiological and biochemical and stability test on them. Then the characterization of NaCl tolerant cell callus line. Then we'll regenerate it on and check that these regenerated plants have that salt tolerance or not. Fine. And then after confirmation, after generations, we'll take them for field performance, and we'll check their seedlings for salt tolerance again. So this is how we perform in vitro selections. Now, detection of somaclonal variants. First of all, different methods of selection and screening. First of all, what we can do, we can go for a morphological characteristic analysis, right? So it could be qualitative and quantitative. Qualitative, we can check height, maturity date, flowering date, leaf size of the plant. Quantitatively, yield of the flower, yield of seeds, quality of seeds, wax content in different plant parts. Okay. Second one is variant detection by cytological studies. Staining of metameristic tissue like root tape, shoot tape, leaf tape with fulgen and acetocaramine provide the number of morphology of the chromosomes. We stain these thin slice of meristematic tissues. We keep it under the microscope and check for the chromosomal morphology for aneuploidy, polyploidy, etc. Right? Then variant detection by DNA content. So cytophotometer detection by fulgen stain nucleus can be used to measure the DNA content. How much DNA? If there is aneuploidy, then there is some difference in the number. Triploidy, right? So there is a difference in the DNA content of the cell. Now variation detection by gel electrophoresis. Change in concentration of enzymes, proteins, chemicals, should we see here, like pigments, alkaloids, and amino acids can be detected by their electrophoretic pattern, by the band, their size, their weight, this, right? Now, detection of disease-resistant variant. Pathogen toxicity or toxin responsible for disease resistance can be used as selection agent during the culture. So this is what we have read in the in vitro selection. We are using a toxin, right, from isolated from a microorganism. Now detection of herbicide resistant variants, plantlets generated by adding herbicide to the cell culture. Now here we will use a herbicide at the place of toxin. Fine, we are using a herbicide and in the cell culture, and then we'll check the resistant cells and we'll grow the resistant variety. Then is the detection of environmental stress tolerant variants. Environmental stress can be salt, water, temperature, or nutrient quality of the soil, right? Minerals like Rajasthan have acidity and basicity, high salt content, like different things, fine? So selection of high salt tolerant cell lines in tobacco, this is an example tobacco. Selection of waterlogging and drought resistance cell lines in tomato. So there is an example like we are making a rice variety which is resistance to drought. Okay, as normally water needs a lot of water to grow. Rice needs a lot of water to grow. So we are making a variety where rice would not need so much of water to grow, right? Now selection of temperature stress tolerant in cell lines in pear. Selection of mineral toxicity tolerant in sorghum plant, mainly for aluminium toxicity. So here is the list of few plants which are somaclonal variants. Let me name some, like tobacco. We just had its name in the example, and the selective agents is methionine, sulfoxamine, right? Like it's pigeon pea, potato, mango, sunflower, lemon. There are so many varieties we have. If you like the video, like, share and subscribe, and we'll meet soon in another video. Bye.